Good morning, everyone. I'm Simran Da Costa, and on behalf of Altum Technologies, it gives me immense pleasure to welcome you all to another session of our Women's Day special webinar series. I'm thankful to all of you for sparing your valuable time to join us today. So, in honor of International Women's Day, which is annually on the 8th of March, this webinar series aims to highlight the accomplishments, dedication, fortitude, and remarkable achievements, commitment, and resilience that women have across multiple scientific domains. In today's session, we will dive into an insightful lecture on drug repurposing, an effective tool to accelerate the drug discovery process. So, before we dive deeper into the topic for today's session, a small introduction to the topic. So, drug repurposing is also known as drug repositioning, and it is a very promising strategy to accelerate the drug discovery process by identifying new therapeutic uses for existing approved drugs. This approach offers significant advantages over traditional drug development, including shorter development timelines, lower costs, and reduced risks. And by leveraging extensive clinical and safety data from previously approved medications, drug repurposing can bypass many early stage challenges such as toxicity concerns and regulatory hurdles. Moreover, repurposing existing drugs can provide rapid solutions for emerging diseases or unmet medical needs, making it an effective tool in the fight against complex conditions like cancer, neurodegenerative diseases, and infectious diseases. During the initial stages of this process, we are now widely seeing the application of advanced computational techniques, high throughput screening, and systems biology that can further enhance the potential of drug repurposing, thereby facilitating the discovery of novel indications and enabling more efficient therapeutic advancements. Today's talk is going to give us an insight and concrete evidence for the potential use of calcitriol, an antiproliferative anti-tumor adjuvant drug in the treatment of ERB2 positive breast cancer patients. We will see the anti-cancer efficacy of calcitriol due to its interaction with the EGFR family member receptors, and we will also look into the explanation on calcitriol that may possess the anti-breast cancer effect through the interaction with all four EGFR family member receptors. So let's understand more about this topic from our speaker of the day. It gives me immense pleasure to introduce Dr. Jubi Selvaraj. Dr. Jubi is an associate professor at the Department of Pharmaceutical Chemistry at JSS College of Pharmacy, UT. She pursued a B Pharm in SB College of Pharmacy, M Pharm in KM College of Pharmacy, and PhD from JSS Academy of Higher Education and Research, Mysuru. She was declared the best outgoing student during her M Pharm. Her experience includes 22 years of both teaching and research. Her research areas of interest are discovering novel chemical entities for cancer therapy through various in silico tools and drug scaffold repurposing. As part of the citizenship activities of the institution and the university, she was a member of NAC and ACPE preparatory teams and is currently a member of the NBA and NIRF teams. She has published 88 international papers and 34 national papers in index journals with a cumulative impact factor of 155.02 in the range of zero point 0.4 to 13.0. Her total citations are 2071 with an H index of 22 on Google Scholar, 17 on Scopus, and 13 on Web of Science. She has been granted 7 Indian patents, and in addition to it, she has published and filed 7 Indian patents. She also has published 5 books and 8 book chapters. Currently, she is carrying out 3 research grants sponsored by AICTE, RPS, ICMR Government of India, and GSS Academy of Higher Education and Research, Mysuru. She has acquired 2 research grants funded by DST India, one international grant from Shakra University, Saudi Arabia, and 5 research grants sponsored by JSS Academy of Higher Education and Research, Mysuru, as a principal and co investigator. She has attended 19 international and 42 national conferences as a delegate. She has been invited as a resource person at 2 international and 21 national conferences and 19 webinars. She's guided 5 PhD students and 14 M.Pharm students, 15 B.Pharm projects, and 5 PhD students are currently working under her. She is a recipient of the prestigious Pharmacy Woman of the Year 2023 from Association of Pharmaceutical Teachers of India Central Branch and Tamil Nadu State Branch, the Young Researcher Award 2019 and Young Performer of the Year 2020 from Association of Pharmacy Professionals India and Best Researcher Award 2019 from GSS Academy of Higher Education and Research Mysuru, Associate Fellow of Antiviral Research Society India, Best Teacher Award 2022 from GSS College of Pharmacy UT. So, without further ado, I'd like to hand over the platform to our expert. So, thank you so much for joining us today, ma'am, and welcome. I invite you to take over the session.
Yeah. Thank you, Simran, for the nice introduction. A very good morning to one and all who gathered virtually. So, at the outset, I thank the authorities and scientists from Altum Technologies Limited, especially Miss Divya, Miss Simran, and Miss Sheetal for giving me an opportunity to share my thoughts on this topic, especially during this Women's Day special webinar series. So, my topic would be a drug repurposing, an effective tool to accelerate the drug discovery. So, in a lighter way, we can say that an old wine in new bottle, since we are playing around with the existing drugs for new intended therapeutic activity. So my presentation goes like this. The first few slides will be an overview of drug repurposing. Then what are all the different approaches used for drug repurposing? Advantages over the conventional drug discovery. Of course, the drug repurposing has some challenges, and I'll be ending up my presentation with a case study where we have integrated 2 to 3 in silico tools and in vitro studies to give up a final candidate. So, according to the Nobelist pharmacologist Sir James W. Black, the most fruitful basis of the discovery of a new drug is to start with an old drug. So, why we are very particular about this drug repurposing? As we all know very clear, the drug discovery process, it is a very complex one. It takes around 15 years for a single drug to come out. Okay, so it has got many phases, starting with discover and development, preclinical research, clinical research, FDA review, and FDA post-market safety monitoring. So around $2 to $3 billion of money is involved. Why we are very particular about this drug repurposing? Why? Because the drug we are taking a de-risk compound, because the drug is already established for its safety profile. So only we can see the intended or efficacy for the other use. Okay, so basically this drug repurposing, no, it mainly follows the principle of polypharmacology. That is, a molecule, it can have the off target effects also. Okay, the on target effect means it has a particular effect, so it will give you a particular therapeutic activity. Apart from that, it may have some off target effects. So due to this off target effects, it may produce some adverse effects and some toxicities also. So what happens? This adverse effect, no, sometimes it may be a significant therapeutic effect also. So it mainly plays, or basically it is from this polypharmacology. So you can see here the time duration, almost 3 to 4 years you can reduce from the conventional drug discovery. And the major three advantages, as mentioned by Ms. Simran in the introductory remarks, the time will be less, the cost will be less, and the safety concerns will be less, and there are chances or less failure in clinic. Okay, so that's what we are very much particular about this drug repurposing. So, what is the proper definition for drug repurposing? The drug repurposing, also called drug repositioning, reprofiling, or retasking. So, it is basically a strategy for identifying new uses for approved or investigational drugs that are outside the scope of the original medical indication. That's what I mentioned. The new uses, it may arise due to the off target effects. Okay, so the original medical indication will be one. Out of that, the drug will be used for other uses. Okay, that will be called as drug repurposing. So, as I mentioned earlier, the drug repurposing is based on polypharmacology, a field of study related to design, discovery, synthesis, and use of pharmaceutical agents. It may be anything, it may be drug, small molecules, antibodies, stabilized peptides, or natural products that act on multiple targets. So, the conventional one drug, one target, no. Apart from that, it will go one drug which act on multiple targets. Okay, so that is basically the principle for this drug repurposing. So the advantages, the first one is the risk of failure is very low, because the repurposed drug has already been found to be sufficiently safe in preclinical models, and so we can avoid the preclinical model safety testing, and the clinical trial safety profile aspects we can avoid. And the time frame, maybe 3 to 4 years, can be reduced, and less investment is needed. So one statistics says that the cost of bringing a repurposed drug to market have been estimated to be $300 billion on average, compared with an estimated $2 to $3 billion for a new chemical entity. Okay, and the regulatory and phase 3 cost, but it will be same, more or less same, okay, for the repurposed drug and for a conventional drug discovery. But still, there will be substantial savings in preclinical and phase 1 and phase 2 cost. Mainly you can get more insights to new targets and pathways that can be further exploited. Yeah, so this is multiple pathways. Okay, again it is mentioned very well by Simran, that is some complex diseases such as neurodegenerative disorders, cancer, so they involve multiple pathways. Okay, so they involve multiple targets. So one such disease is rheumatoid or arthritis. As an example, I am showing here. So what happens during this RA? There will be these pro-inflammatory cytokines will be overexpressed. Okay, in turn they activate so many pathways: notch signaling pathway, SVK, P13, JAK stat pathway, WNT signaling pathway, MAPK signaling pathway. So one such pro-inflammatory cytokine is IL-6, interleukin 6. Okay, so it activates the JAK stat pathway, that is Janus active kinase and stat signal, the stat pathway. Okay, so these two expressions, JAK and stat expressions, no, it will be very they will be like abnormal in this rheumatoid arthritis. Okay, so in turn they will induce some important protein, especially this mmp matrix metalloproteinase expression will be more. Okay, so due to that, the accumulation of chondrocytes in the synovial joints, what I am trying to tell is it involves multiple pathways. Okay, not what non it is not unified or not particular to a single pathway. Okay, so here the drug repurposing or the off target effects, no, it will give you some ideas, okay, for the new intended therapy. So one such example is the sildenafil. Okay, so the sildenafil is originally it is intended for anti-hypertensive property. It is a very good anti-hypertensive drug. Okay, but as an on off-target effect, okay, so it is producing or it is used to treat male erectile dysfunction also. So this particular off-target effect paved the way the sildenafil know for very blockbuster drug. Okay, we know very well it is Viagra produced from Pfizer. Okay, it is a very blockbuster drug which is exclusively used for this erectile dysfunction. Okay, so the on-target effect is the anti-hypertensive property, but unfortunately, we can't tell it is unfortunate. It is a fortunate. So it is used for now off-target effect. It is used for male erectile dysfunction. And another one example is thalidomide. Okay, it is a sedative originally marketed in some countries in 1957. Okay, so it was it is mainly used for the pregnant woman. Okay, so it was withdrawn within 4 years owing to its infamous link with severe skeletal birth defects in children born to mothers who had taken the drug during the first trimester of their pregnancies. However, so after the drug was withdrawn, it was serendipitously, that's what I told, it is opportunistic and serendipitous, the drug repurposing. Okay. So it was found to be effective first in the treatment of erythema nodosum leprosum. Okay. It is a very complicated condition in leprosy, which is immune related disorder. Later on, it is used for the treatment of multiple myeloma also. Okay, so these paved the way. These off-target effects, no. Okay, so now these drug and its derivative, which is called as lenalidomide, these two drugs are very much useful in this ENL, that is erythema nodosum leprosum, as well as the multiple myeloma. So this is the successful stories of drug repurposing. Okay, so the list goes on. Zidovudine, the original indication is for cancer and the new indication is for HIV and AIDS. It is approved in the year of 1987. Likewise, minoxidil, it is hypertension. Now it is very much useful for the hair loss treatment. Then sildenafil, just now we have seen, it is used for erectile dysfunction. Thalidomide, original indication is it is useful for the treatment of morning sickness for pregnant women, especially for first trimester. Now it is used for ENL and multiple myeloma. Celecoxib, it is a very good COX2 inhibitor, okay, but it is now used for familial adenomatous polyps treatment. Okay, likewise, you have the list, it is going on. Yeah. Now we are moving to the second part of our presentation, the approaches. What are all the approaches you can choose for drug repurposing? So any approach you can take, it involves the very basic three steps. The first step is identification of a candidate molecule for a given indication. Okay, so you have to identify a particular molecule. So here, this step will be very, very critical, I say, because the hypothesis, you have to generate the hypothesis in a with a proper justification. Okay, so here the modern approaches, all the in silico approaches can be useful. Okay, the publicly available databases can be useful for the generation of this hypothesis or picking up that particular drug. The second step is mechanistic assessment of a drug effect in preclinical models. Okay, once you identify a drug once you prove the drug will be useful by suitable in silico drug discovery techniques. Then we have to move for in vitro as well as preclinical models. And the phase 3 is evaluation of efficacy in phase 2 clinical trials. Okay, so here we assume that there is sufficient safety data from phase 1 studies. Already the drug, it may be proven for its safety data. Okay, so we can skip that and directly go to the phase 2 study. So these three steps will be very very basic for any approach you choose. Basically, you can divide the drug repurposing into two categories. One is in silico drug repurposing. The other one is activity based in silico repurposing. Okay. So in silico repurposing, the very basic example is structure based drug design. Okay. One example I am taking here. Okay. So if you want to find out a drug for a particular intention. Okay, so what you can do, you have to go through the disease pathway of a particular disease. Okay, you have to go through the molecular pathways and you have to finalize one particular target. Okay, so then you have to predict the structure of a particular target by maybe x-ray crystallography or NMR technique. On the other hand side, no, you have a database, okay, existing drugs. From this only you are going to pick up a particular drug against this particular crystallography protein. Okay, then you can do a simple technique, molecular docking. Then you can inspect the docked poses, then potential compounds. Then finally you can validate with molecular dynamic simulation, and you have to go for in vitro analysis, preclinical studies, and finally, yeah. So this before this in vitro and preclinical studies, no, we have the hit compounds. Okay, so here this in silico repurposing, it majorly utilizes the publicly available databases, both bioinformatics and chem informatics. Okay, and you can do some network construction. You can construct some network with the protein protein interaction and protein drug interaction. On my right hand side, it is purely, it is activity based in silico repurposing. Here the physical, the actual drugs may be required for the screening. It may be for anti-cancer activity. If example, you will take MTT, you will do MTT assay, flow cytometry for cell cycle analysis, and in vivo preclinical studies. This is the broader classification of drug repurposing. Again, these are the various approaches for in silico repurposing. Okay, so start with signature matching, where the signature, it may be the transcriptomic signature or structural chemical signature or adverse effect signature. It may be compared with drug or drug disease. And the second one is the molecular docking. Okay, it is a structure based computational strategy, and there will be genetic association studies. Okay, a particular variant or particular mutant will be compared for both drug and drug or drug and disease. And this is pathway mapping. It is a well known and very commonly used for this drug repurposing. You can construct the networks, that is network analysis, using genetic, protein or disease data. Then you have clinical analysis, retrospective clinical analysis, mainly electronic health records. Okay, adverse effects data, you can make it and you can compare. Okay, so these are all the in silico repurposing approaches. Okay, and the down one is your in vitro technique, the experimental approaches, phenotypic screening or binding assays. Yeah, let us quickly move what is what. The first one is the computational technique, that is signature matching. Okay, so signature matching is based on the comparison of the unique characteristics or signature of a drug against another drug or disease or a clinical phenotype. So here the signature, it may be transcriptomic signature, proteomic or metabolomic signature, it may be a chemical signature, or it may be a adverse event profile. So basically you are comparing the signature profile between two drugs or drug and disease. So the first one is matching transcriptomic signatures. You have two types: drug disease similarity and drug similarity. So the first one is here, the transcriptomic signature of a particular drug is derived by comparing the gene expression profile of a biological material such as cell or a tissue before and after the treatment, the after treatment with the drug. So basically here you are comparing the gene expression profile. Okay, so this differential gene expression signature is then compared with a disease associated expression profile that will be compared with the differential expression analysis of disease versus healthy conditions. Okay, so the very good example is this topiramate. Okay, so this topiramate is an anti-epileptic drug. Okay, so what actually they wanted this topiramate for another therapeutic indications also. So especially they wanted to find out some drug for inflammatory bowel disorder. Okay, so for this what they did, okay, so they have used the publicly available data, that is NCBI gene expression omnibus. So from this they have taken the transcriptomic signatures of the disease. Okay, then there is one connectivity map. Okay, it gives the gene expression profile of 164 drugs. Then they have matched the transcriptomic signature of both. From that they found out this topiramate, okay, which is now used very well for this IFD disease. On the other hand, drug similarity, again here instead of drug disease, here they have compared the transcriptomic signature between two drugs. Okay, so and here what is the basic understanding is the shared mechanism of action will have the similar type of therapeutic activity. Okay, here it is not necessary that they should be structurally similar. Even the structurally dissimilar compounds will also have they also share the same transcriptomic signature. So originally this fasudil, it is basically it is a rho kinase inhibitor, rock inhibitor. It is now used for this neurodegenerative disorder, that is amyotrophic lateral sclerosis. And this is computational molecular docking. So normally in conventional docking, what we do, we take a particular drug, okay, and or a particular drug, and we will dock with multiple targets. Okay, so then we find out which one is used, all those things. Okay, here this conversely, drug libraries could be explored against an array of target receptors. Okay, several targets and one ligand. Okay, usually in normal conventional docking, no, we take one target and multiple ligands, correct? So from this multiple ligands, we choose which ligand is very much suitable for that particular target. So here we go for several targets, okay, and the drug which is against which may be suitable for which target. Okay, so the very good example is mebendazole. So mebendazole is an anthelmintic drug. Now it is used for this inhibitor of VEGFR2, which is a very good mediator of angiogenesis. This is our another technique. And another technique is GWAS technique, genome wide association studies. So this GWAS, it is basically based on the genetic variants. Okay, so they identify which variant or which mutant is associated with a particular disease. Then that variant, it may be associated with some other disease also. So basically they deeply they will look into the genetic variants. Okay, so this is again the example is denosumab. It is used for the treatment of postmenopausal women with high risk fracture of osteoporosis. The target is tumor necrosis factor superfamily member TNF SF11, which is also called as rankle factor. So here, one of the variant, which is called as RS2062305, this variant is majorly involved in this particular disease. Accidentally, that variant is also found out in Crohn's disease also. So it is used for the treatment of Crohn's disease also. Then the next one is pathway or network mapping. So here you have multiple genes, okay, so or multiple proteins. You can construct the network with the multiple genes or multiple proteins. That is one protein protein interaction. The same thing, you can construct the network with protein and drugs. We call it as protein drug interactions, PDI networking. So this is very much useful in this field. So the pathway based or network based approaches have been widely used to identify drugs or drug targets that may have potential in repurposing. So the network analysis involves constructing drug or disease networks based on gene expression patterns, disease pathology, protein interactions, or GWAS data in order to aid identification of repurposing candidates. So here you can see this pranulocast and amrinone. Okay, so this pranulocast is a leukotriene receptor 1 antagonist which is used in asthma, and another one is PDE inhibitor amrinone. Okay, so this is used in congestive cardiac failure. So both of these drugs now emerged or they are studied for the treatment of viral infection. That means they, what are all the genes associated with this viral infection, so they are networked with these two drugs, pranula with the no with variety of drugs. From that these two drugs have come out, okay, for the treatment of viral infections. And using electronic retrospective clinical analysis, use of electronic health records. So the retrospective clinical data can be obtained from various sources including EHRs, post marketing surveillance data, and clinical trial data. So the EHRs contain an enormous amount of data on patient outcomes, both structured and unstructured. So the diagnostic and pathophysiological data, including the results of laboratory tests as well as the drug prescribing data, are more structured. So from this data mining, okay, we can go for the repurposing. Example, terbutaline anti-asthmatic drug for the treatment of amyotrophic lateral sclerosis. So these are all about some in silico techniques. And coming to the experimental techniques, the very well-known technique is the binding assays to identify the target interactions. Binding enzyme binding assays. Okay, proteomic techniques such as affinity chromatography and mass spectrometry have been used as approaches to identify binding partners for an increasing number of drugs. One such technique is SETSA technique, cellular thermostability assay. Okay, here imatinib is invented or found out for gastrointestinal stromal tumors. The other one is phenotypic screening. The phenotypic screening can identify compounds that show disease relevant effects in model systems without prior knowledge of the targets affected. Okay, we do not know anything about the target. It is very basic for high throughput cell based screening. So, these are the two techniques. Now, this message is very, very important. None of these methods alone will be sufficiently able to disclose the complex interplay between drugs, targets, and disease. Okay, so what I wanted to convey here, we have to integrate one or more computational approaches. Okay, so to navigate through the wealth of available information, and hopefully we can find clues solid enough to justify a repurposing hypothesis worth of experimental investigation. Okay, we can pick up 2 to 3 in silico techniques, and we can prove our hypothesis by doing this in silico experiments, then we have to validate the in silico data with the experimentally available screening methods. That's what we have to integrate the various tools. So again, the choice of the most appropriate methods will basically depend on the nature of the problem to solve and on the type, quality and quantity of information. Okay, so whether you can you can go for structure based molecular docking studies, or you do the networking analysis, or you do signature matching, transcriptomic matching, you can do the genetic variant study. So what you want, accordingly you have to select the particular tool, and very importantly they are all dependent highly on the public or proprietary databases. So this is one such interesting data site. Okay, drug repurposing online tool, and these are the various databases. Okay, where you can collect the structures. For example, if you want the chemical structures, you can go for PubChem. You want the drug, you can go from, you can take it from drug bank. Okay, then for target three-dimensional structures, you can very well rely on RCSB, that is protein data bank. Likewise, you have some data bases. From that, you can collect the drugs or the targets, that is protein structures. Of course, the drug repurposing also has some limitations. Okay, so the pathways and mechanisms of many complex disease remain incomplete. Okay, so that is a major struggle. And the second one is the data. Well, we need well curated data. Okay, sometimes the database, sometimes it may mislead. That is the second drawback. And there is compared to conventional drug discovery projects, the fundings are limited. There is no systematic mechanism to obtain fundings to support drug repurposing projects. I'm not telling it is not there, but relatively it is less. Of course, we have the patent issues, market exclusivity issues, that is new method of use patent. MOU can be obtained for a new purpose use of an old generic drug, and we do have some regulatory changes. Yeah. With this, now I am going to explain a case study where we integrated 2 to 3 in silico tools, and we have validated that study with the in vitro study. Okay, it is an in silico drug repositioning approach of identification of drug candidates for breast cancer therapy. It is a study which is carried out by one of my research scholar, Dr. Naharaj. So our hypothesis, that is the goal of this research work, is to identify the drugs for the suitable targets of breast cancer pathways through in silico drug repurposing approach. Okay, so we collect the information such as drug, target, proteins, genes and pathways. Then we do construct the network like drug similarity, target similarity network. Then it goes for random walk prediction. We predict the drug target interaction based structure based analysis. Then we identify the suitable drug candidate. If it is on patent, use as it is. Okay, off patent, again use as it is, like publicly driven development. So this is our basically our hypothesis. So with this, our aim is to identify the drugs for the suitable targets for breast cancer pathways through an in silico drug repurposing approach. We have 3 objectives. Our first objective is to identify the suitable targets for breast cancer therapy and suitable drugs for selected targets through molecular networking. The first tool we use here. The second one is, from this, to select suitable drugs for the selected targets by combining structure based molecular docking and molecular dynamic simulation approach. The second in silico approach we use here. And third one is to evaluate the selected drugs for their target specific anti breast cancer activity using suitable in vitro method, that is the experimental technique used for drug repurposing. So these are the various databases we have used. As I told you, this in silico technique is completely dependent on the databases and softwares. Okay, these are the databases on the right hand side. These are the softwares what we have used, both like online tools as well as the Desmond and from Schrodinger. We have used the commercial available softwares also and freely online tools also. Majorly, we have used the online tools. Okay, and this is the database what we have explored. So coming to our first objective. Okay, so we have divided into five clusters. The first one is selection of target. We have to select the target. Then after the selection of targets, we constructed the protein-protein interaction network. And cluster maker is narrowed down from the sub network formation from the PPI network. Then we have constructed the PDI, protein drug interaction networks, and finally we selected the drugs. Yeah. So, as I mentioned in my title, our intention is to identify a suitable candidate or a drug molecule for a breast cancer. Although estrogen receptor pathway is the primary pathway altered in breast cancer, it is now well accepted that many other signaling pathways are also involved in the pathogenesis of breast cancer. So, therefore, understanding these additional pathways are essential to explore the new therapeutic approaches for breast cancer therapy. So, these are all the other pathways which are involved in this breast cancer: EGFR overexpression, P13K, MAPK, WNT not signaling pathway. So, majorly we are concentrating on this EGFR overexpression signaling pathway. Yeah. First study we have collected what are all the various genes which are related to this EGFR pathway. The table shows the various genes. Okay. Then from this genes we wanted to narrow down. We wanted to cut down. There is a tool called top gene suit. Okay, so it is basically for the gene enrichment or prioritization analysis. Okay, we have one score, average score. So this average score is based on various pathological factors. Okay, so it is a relative factor. So we have we have tabulated the score. Okay, and based on these average score, no, which is more than zero point 0.7. Okay, almost 0.69 to 0.77. So we have considered this many genes. The 50 genes have been narrowed down for our further study. Then we have constructed the molecular networking. Okay, protein-protein interaction network. Then cluster analysis. We have to characterize the PPI network. Then from that, we have to collect the suitable target. Then we have to do the protein drug interaction. Before that, we need to collect the drugs from various databases. Again, you have to characterize the PDI network. Then finally, you can identify the drugs. Yeah. So the 50 genes are collected, okay, from the top gene suit analysis. From that, the network is constructed. The PPA network of the selected genes was generated by using the string protein query of the Cytoscape 3.8.2 software. So here, the genes can be considered probable only when the confidence score is greater than or equal to zero point 0.4. So the genes were constructed, the network is constructed among the 50 genes. Okay. So you can see here, this network has got 50 nodes. The node is nothing but the gene, the round. Okay. And the edges, there are 527 edges. The edges is nothing but the connection between two nodes, that is called as edge. So this is node and this is edge. Okay. This network can be analyzed. Then it has to be characterized very importantly. Okay. So for that, we have used the CytoHubba zero point 0.1 app plug. Okay. Basically, these are the three important parameters we look into. One is degree. Okay. closeness centrality, and betweenness centrality. Okay, so these three parameters will tell you how a particular node is connected. Okay, if the degree means the degree is more, means the particular node has more connections in the network. Okay, betweenness centrality means how closeness centrality, how within a short period of time how many nodes it can connect. Okay, likewise it will tell you some parameters, okay, physical parameters. So these three are very very important. And afterwards, no, from this major network, no, okay, so then we constructed sub networks. For that we have used the M code app. It is mainly used for cluster maker. Okay, so this cluster maker, from this it has been clustered, two clusters have been made, okay, based on the functional similarities between the genes. So the cluster one is little big. The genes which are very much associated with cancer and which are very much associated to breast cancer. The cluster 2 is associated with cancer. Other than cancer, these genes are also associated with other diseases like liver cirrhosis, diabetes mellitus, cardiac disease, and CNS disorders. So this is the cluster maker or sub network construction. Okay, so this is the table which give you the scores of these clusters. You can see here the cluster 1 has got 24 genes, okay, and 251 edges, okay, the connections, and these are all the very important, the 24 genes. And coming to the cluster 2, it has got four genes, four, sorry, seven, score is 4, but 7 genes it has got and 12 edges. Okay, and this is the physical parameters, okay, degree, betweenness centrality, and closeness centrality for the cluster 1. You can see here almost there is 9 genes which are having more degree. That is, it has got the degree, that means it has got more connections. Okay, that means it is more related to the EGFR pathway. That's what it is telling. Again, the betweenness centrality also based on these, no, the EGFR family, that is EGFR, ERBB2, ERBB3, and ERBB4. So these further 4 genes have been taken for constructing the protein drug interaction network. As I told you, for construction of protein drug interaction network, no, we need the drugs. Okay, so the drugs have been collected from drug bank, pubchem, and zinc 15 databases. So around 2637 drugs were collected. Then the network is constructed, okay, between the 4 genes with this 2637 drugs. Okay, and these drugs have been segregated into 4 categories. The first category is the drugs indicated for breast cancer therapy, drugs indicated for any cancer therapy other than breast cancer. Third category is drugs indicated for non-cancer related disease or disorders, drugs contraindicated for breast cancer patients. So among these 4 category, the third one, drugs indicated for non-cancer related diseases, are given more attention for our further study. So we have constructed with that third category of drugs. Okay, this is the first table which gives the topological measures like degree, betweenness centrality, and closeness centrality of the PDA network with ERBB2 gene. This is EGFR gene, and this is for ERBB4 gene, and this is for ERBB3 gene. So here we constructed 4 networks, okay, with the 4 genes have been finalized, and the drugs which are indicated for other non-cancer related disorders. Okay, and coming to our second objective is the structure based molecular docking and molecular dynamic studies. We have finalized the target now. Now we have to select the suitable drugs by using molecular docking, MMGB assay, molecular dynamics, and MMPB assay. And these are all the very very basic protocols, so we know very well. We have to identify the three dimensional structures for all our targets based on these various physicochemical parameters like binding site and theoretical pi. So these are the four protein data bank ideas. Four proteins finally we have identified: EGFR, ERBB2, 4, and these are all the PDB IDs. And the molecular docking studies have been carried out. Okay, and all the drugs know it has been compared with the co-crystal of that particular protein. And one more additionally, ERBB2 positive breast cancer drug neratinib has been taken as a standard for comparison of the molecular docking results, the binding affinity results. Okay, so this is the result for all the four compounds, four enzymes: EGFR, BB2, BB4, and BB3. So the calcitriol, no, among these all the drugs, no, it is showing good binding affinity compared with co-crystal and neratinib. Okay, so we have taken this calcitriol, okay, for our in vitro study. And not only that, we have analyzed the interactions, especially the non-covalent interactions, okay, like hydrogen bond, hydrophobic, all those interactions we have compared. And finally we have validated with mmgb assay, okay, to analyze and calculate the binding free energy, to ascertain the binding stability of the complex. And our observations were the results of mmgb assay showed that the binding of calcitriol with the 3-RCD and 1xkk proteins was relatively stable. The coulombic energy among the various binding energies like van der Waals, hydrogen bond, covalent, lipophilic, which favors more the electrostatic coulombic energy source the favor. It is validated with we check the stability by 100 nanoseconds run of molecular dynamic simulation also. Followed by that, we did the MMPB assay binding free energy also. Okay, and we come to a prayer conclusion, and we picked out the calcitriol compound, okay, which is showing a very good very good results, okay, in all the four structure based in silico studies. Now, moving to our third objective, we have picked out a drug, moiety, okay, which should be validated by using available in vitro methods. So, for that, you have used 5 different studies: MTT assay, apoptotic signaling pathway, scratch wound healing assay, cell cycle arrest by flow cytometry, gene expression study. For all these, the results are compared with the neratinib. Okay, neratinib was used as a standard. So the first study is cytotoxicity by using MTT assay. Here we have used Vero cell lines to ascertain the safety profile. Okay, the toxicity is very high in Vero cell line, whereas in MCF7 cell, which is overexpressed for ERBB2. Okay, so this is you can see here calcitriol is relatively showing the value close to the neratinib, that is 44.35. So from this we may have the interference that the calcitriol possess it may have comparable cytotoxicity, and it decreased the proliferation rate of breast cancer as like standard neratinib. Then it has been moved further for intrinsic apoptotic signaling pathway. We did some 2 to 3 studies under this section. The first study is acridine orange and ethidium bromide assay. Okay, so this study tells that the calcitriol induces the apoptosis in breast cancer cells, further supported by HOE, CHST, nuclear apoptosis training, and rhodamine staining. Okay, then the second one is cell migration ability. Okay, by using scratch wound healing assay, the wound has been created, and the drug has been administered. We have seen the cell migration ability. Okay, so the after administering the calcitriol and neratinib to the cells, that is MCF47 cells, the observation was taken at different time intervals like 0 hour, 24 hours, or 48 hours in order to determine the proliferating ability. Okay, so you can see here the control, no, the wound has been closed. Okay, the wound width is little bit compared to 0 24 hours, it is less, and 48 hours it is completely closed. But in calcitriol and neratinib, no, the wound width, it is like that only, there is no significant closure. Okay, so it tells that it may have the significant effect on the proliferating, sorry, the proliferation inhibiting capacity. And this is the wound with statistical representation. Then we have checked the cell cycle analysis, which phase the calcitriol may have the effect. Okay, so the calcitriol treated cells showed the increased percentage of G0 and G1 phase cells when compared to the standard neratinib. And finally, we ended up with this gene expression study. These are the basic protocols like RNA extraction, cDNA synthesis. Okay, then we have calculated the delta delta CT values, and our observation is, the final observation is, it is significantly downstreaming the ERBB, the EGFR signaling. So, coming to the summary, the present study implies a systematic drug repurposing approach for breast cancer therapy. So here the process initiated with the step of collecting the genes related to EGFR overexpression signaling pathway. Around 700 genes were collected. From that we have narrowed down 50 genes based on the top gene, based on the average score. From the PPI network we finalized 4 targets. And from the PDI network, these many drugs were identified interacting with these receptors. And from these four, we have segregated 4 categories. From that, the drugs approved for the treatment of non-cancer related disease disorders were focused. And finally, they have done for the structure based studies, molecular docking, and MMGB and MMPB assay, and MDC simulation also ascertain the stability of the complex, calcitriol complex with the with the proteins. Then we need to validate the in silico studies with the in vitro studies. So in vitro studies also revealed that calcitriol has shown significant cytotoxicity and decreased the proliferation rate of breast cancer cells as like that of standard neratinib without causing any harm to normal cells. And finally, these studies are finally confirmed by apoptosis studies, scratch wound healing assay, cell cycle arrest analysis, and gene expression studies by showing the similar efficacy to that of neratinib. Coming to the conclusion, concretely, the overall results of this study provides the concrete evidence for the potential use of calcitriol for it may be repurposed for the treatment of antiproliferative, antitumoral drug in the treatment of ERBB2. However, it should be further validated with further studies in vivo, preclinical studies. Okay, so it may be further explored clinically for breast cancer therapy, provided it passes all the safety and profile. So with this particular work, we have generated 3 publications. Okay, the first one is the pure in silico work, and this one is anti-cancer agents in medicinal chemistry. The in vitro study has been published. And before starting, we have completely reviewed network based drug repurposing. And I acknowledge my management, that is my pranams to His Holiness Swamiji of JSS Suturmat, and my thanks goes for all the leadership of JSS Academy of Higher Education and Research, principal and management of JSS College of Pharmacy UT, and always my credit goes to my thanks goes to my PhD and PG students. And this work is carried out by, as I mentioned earlier, one of the research scholar. And I have published a book for this computer aided drug design. It is basically for final year B pharmacy and M pharmacy students. Of course, other bioinformatics and life sciences related streams also can refer this book for your needs. So once again, I thank the people from Altum Technologies Private Limited for giving me an opportunity to share my experiences on the topic drug repurposing during this Women's Day special webinar series. Thank you one and all. Thank you again. Yeah.
So thank you so much, ma'am. This was a very informative, very detailed, and very well explained session. So I'm sure that some of our viewers have some questions. So if you have any questions, guys, please post them in the chat. So the first question is how to find active site in target protein and docking result analysis.
Yeah. So again, if you are, it is again varies from software to software. Okay, so basically if you have the co-crystal, okay, so based on that you can predict the binding site. Okay, you will be having some options what software you are using it. If you don't have that, you should have what are all the important residues, okay, involved in active site. Based on that, you have so many options like sitemap analysis. You can predict the active, you can design or predict the active site.
Okay. Next question, please. How to uncover new mechanisms of existing drugs using computational methods.
Yeah, so it's a good question. Before going to any tool, you should have a proper literature review. That is very important. Okay, so if you are working for a particular disease, you have to first of all study the pathways. What are all the pathways you have? With that knowledge, no, you can go for network analysis. Okay, you have 4 5 pathways. One by one, you see. First, you select one particular pathway like EGFR pathway, what are all the genes associated with that. Okay, then you go for the second one, then like that. But without basic literature study, you cannot go for computational methods. Okay, you should have a prior knowledge for assigning or doing any work.
Next question, please. What's the difference between signature profile of a drug and molecular fingerprints of a drug?
Signature profile of a drug, it may be two aspect. It may be a RNA transcriptomic signature, or it may be a chemical structure aspect. Molecular fingerprints of a drug, it is basically based on the molecular features, pharmacophoric features. Okay, chemical structure is different, your pharmacophoric feature is different.
Okay, thank you. Do we have any other questions?
I think I have answered this.
Yes, ma'am. Is there another question? Okay. Want to know regarding data collection and pre-processing for training the model and details about where to get data set and how to get access to them.
Yeah. So again, this whatever I have shown, no, majority 70 to 75% they are all publicly available databases only. Okay, and again, if you are using any database, okay, the first cited literature will be there for using that particular database. For example, if you are using Swiss ADME database, there is a paper which is published very first. Okay, so it will give you all the details how to use it. Okay, so it will give you in a stepwise procedure also. So it is again, it is available publicly.
Okay. Do we have any more questions? For molecular dynamic studies,
Yeah,
which
we have
system is
used,
used?
yes, yeah, we have used Desmond module of Schrodinger. We have used that.
Okay. Yeah, I think we are done with the questions. So, yeah, thank you so much, ma'am, once again for coming here and answering the questions and for the great presentation.
Thank you.
Yeah, so actually, it's time for a certificate of appreciation. So, yeah, thank you so much once again, ma'am, for giving your
Yeah,
time. And it was really a pleasure to have you with us. Like, we're very grateful you could join with us today.
thank you. Yeah, have
Thank
a
you so
great day.
much.
Yeah,
Yeah, also, I'd like to thank all of the participants and viewers for joining today. And if you guys want more such sessions, please leave a like, please subscribe, and please follow. So, yeah, thank you so much, everyone, and have a great day. And thank you, ma'am. Have a great day.